Okay.
Yeah, we can see it.
Move them. Yeah. I know. Move the mouse. Yeah, is this in? Yes. We see. Yes.
Okay.
I was looking at my mouse.
So thank you for the invitation. My talk will be very different from the one that we just heard. In particular, there is not going to be any reaction networks or any autocatalysis, which I apologize about. But hopefully it's still going to be interesting. So what I'm actually going to be talking about is also something that's not published yet. So this is a work in progress. And it's basically a pitch to say that we have a particular type of problem, and there is a type of model that is used in a completely different area. And we think that they go together very well, and that this type of model can be used to solve the problem. So my slides tend to be a little chaotic. So of course, if there's something you don't understand, feel free to interrupt me. And basically, the talk is split into 3 parts. First, I'll introduce the problem, then I'll introduce the model, and then I'll put them together and try to persuade you that it's really a good fit. Okay. So our problem concerns microbial communities, and in particular, how their dynamics, so how they kind of stabilize in some configurations, etc. So when I say microbial community, I'm a community scientist. So for me, microbial community is basically, there are some little guys strewn in somewhere. This could be anywhere. This could be human gut. This could be hot vents at the bottom of the ocean. This could be roots of the plant, whatever. And the important point is that my little drawing here is maybe not illustrating this very well, but we actually are interested in the populations of these different bacterial taxa or whatever they would be. So it's not just individual species, individual organisms, but the populations of these organisms. And it has been shown that dynamics of these microbial communities, so how they shape themselves and their microenvironment, depend on how they interact. So for me, the interactions are going to be super simple. We are going to have positive and negative interactions. So the positive would be the green pointy arrows, the negative would be the flat red arrows. And generally, these kind of interactions happen at the level of the metabolic exchange. So there would be some compounds that the species synthesize, then secrete, then a different species can uptake from environment and use in their own metabolism. But this also means that it's very hard to detect these kinds of interactions in the lab. So instead, we want to predict them in silico, computationally. And right now, most of the methods that go in this direction are actually statistical methods. So these methods do not consider at all the dynamics of the system, which, I mean, of course, the hypothesis can still be relevant, but isn't quite as exciting. Now, another option is to go down on the level of the metabolism. So as I mentioned, the exchanges happen on the metabolic level. So methods like flux-balance analysis have been applied. But the problem is that to reconstruct the metabolic networks of all these involved microorganisms, one typically goes to genome scale automatic reconstruction of genome scale models, and these tend to be extremely poor quality. So using flux-balance analysis actually requires a very, very intensive manual curation of the metabolic models. Finally, of course, one can use population models or dynamical models directly on these microbial populations. Typically, this would be an ODE, but also probabilistic models have been applied. But these models suffer from the fact that you need precise parametrisation, which kind of reduces the predictive ability. Right. So this is the problem domain. So what we want to do is we want to predict how these little guys interact in their little communities. And the model that we want to use is Boolean networks. So Boolean networks come from a different area of systems biology. they are being used almost exclusively to model gene regulation. And Boolean networks are a discrete dynamical model as opposed to the continuous models. So they obviously involve some manner of abstraction. And in the Boolean network, the Boolean abstraction is very heavy because we only consider variables that are valued 0 or 1. Nevertheless, Boolean networks have been shown to be able to express very complex dynamical behaviors. So it's not like we are losing out there. Now, formally, the Boolean network is basically just a function that takes a vector of n variables, Boolean variables. So it's a Boolean vector of length n, and spits out another Boolean vector of length n. Now, this is not very practical. So usually we look at the function piecewise and talk about so-called local functions for each of the variables. And the local functions can then be specified in terms of propositional formula, basically. So what this reads is that the variable 1 in a given state x, the state x being the value of each of the variables as a vector, wants to be the opposite of variable 2, etc. Now, looking at the functions is still not very intuitive. So typically, we would depict the networks in terms of interaction graphs. So this is literally what we've basically seen before with the microbial community. There are pairwise interactions between our variables. And you can see that there's a correspondence here. A negative interaction would mean that the variable appears as a negated literal, while a positive literal will appear as a positive literal. Now, of course, this assumes that these functions are somehow minimal and that there are no superfluous elements in them. Right, but the intuition is right there. And the nice thing is that, as I mentioned before, Boolean networks are a discrete dynamical model, which means that we do have dynamics to this interaction graph, right? So interaction graph is an abstraction of this Boolean network because we lose the information on what kind of symbol is here. Right, so the dynamics of Boolean networks are, as is typical for discrete dynamical systems, a transition system. So we will have nodes that are these vectors of Boolean, these Boolean vectors of length n, and between them we will have some semantic relation that gives us the transitions. Well, with Boolean networks, there have been many different semantic relations considered. One of the simplest ones would be so-called asynchronous updating, in which case each variable updates separately. So in each update step, we will choose non-deterministically one variable, and we will update the value according to these functions. Right? So in this case, these dark squares would be zeros. So this would be variable 1, variable 2, variable 3. And we can see that we can choose to either update variable 1 because it wants to differ from variable 2, but the same goes for variable 2, so we can update that one as well. And then in this case, we can go further and update variable 3. So very simple reachability from state 0, 0, 0 here. We can immediately see that this actually is a multi-stationarity because what we have
here white is active and green is 0. Yeah, okay.
Yes, indeed.
And you choose arbitrarily, so to say, how to start from the free.
Yeah, it's non-deterministic. So there's no rules, there's no probabilities, nothing. Yeah. Right. So these so-called fixed points, so configurations from where there is no more updates possible, are interesting for us because they represent the stability of the system. Right. So if you looked at microbial communities, this would mean that the community has stabilized somehow with only one species present. Right. Well, this is just one way to construct Boolean network dynamics. People have also considered, for example, the kind of the opposite, the synchronous updates, when all of the variables update at the same time. And this actually results in a deterministic semantic. I'm not going to go through it though, because there's a lot more. People have considered a lot more than this. And in a way, for a long time, the all-encompassing semantics of Boolean networks were so-called generalized semantics, where we choose any subset of variables and update them in the single step. Right? So in this case, we extend what we had before by being able to update the first two variables, 1 and 2, in one step together. Right? So we get this little loop. So it becomes immediately obvious that to talk about the fixed points as kind of a long-term terminal behavior, we need some notion of fairness, but that's just a kind of obvious notion there. But although for a long time, these kind of generalized semantics were considered to cover everything, it shows that this is still not enough. So if you consider this model on a continuous level, so now we forget that we are abstracting to Boolean, and we think of each variable as being able to have continuous evolution, then we could imagine that both 1 and 2 start increasing, and we could say that maybe this interaction from 2 to 3 is very sensitive. It has a low activation threshold. Meanwhile, this interaction from 1 to 3 is it's not very sensitive, or the 3 is not sensitive to the presence of 1. So 1 has to grow very high before this interaction activates. So it has a high activation threshold. So in such a case, 1 and 2 would both start increasing, and 3 will start increasing almost immediately after 2. And it will have enough time to increase to value 1, or something that will be binarized to 1, before 1 would push it back down. So in a continuous world, we would actually be able to see a state in which all 3 variables are active. And the inability to see the same state in these generalized semantics has led to introduction of so-called most permissive semantics. And they are exactly, I'm not going to go into the technical details. I also think I will go over time, which I apologize for, but basically they are exactly based on allowing any combination of these kinds of activation thresholds. And the nice result from this is that they come with a guarantee that any continuous behavior is captured at the Boolean level. And in particular, this relates to continuous refinements. A continuous refinements meaning that the continuous and the Boolean model agree on variable tendencies. So if there's positive derivation, then there would be the function would want to go to 1. If there's negative derivation, then the function would want to go to 0. Now this looks a little bit like a jungle here. You can see though that we can get the state 1, 1, 1, but you can also see that the multi-stationarity is maintained. There's one steady state here, one fixed point here, and the second fixed point is here. Right, so there are still the 2 states that we cannot leave anymore once we reach. Right, so this is the brief introduction to the model, right? And I think it's immediately quite obvious that if we have a model that's basically interacting entities, and we want to model interactions between entities, then this is a good fit, or at least theoretically it should be. So to confirm this assumption in practice, I should say, before I continue, that there's, of course, the question of interpretation. Right? So I said before that we are interested in populations of microbes, of bacteria, and now we have Boolean variables. So what does 0 and 1 mean on a population of microbes? Well, obviously, the kind of natural way to say would be absence presence, but that would yield very boring dynamics because 0 could never change again. If something goes extinct, it's extinct. That's it. So instead, we are a little bit more hand-wavy with it, and we say that if a population is large enough, if the species is abundant, then the value 1 means that it actively influences the microenvironment around it. So its interactions are active, so it's 1. And if it's sparse, probably doesn't have that much influence on its microenvironment, so that it will be 0 and the interactions are inactive. Right. Okay, so to confirm that this works, I was quite lucky that this paper has been raised to my attention by Jonathan Friedman and friends, where they picked 8 different bacterial species, bacterial taxa. I honestly do not remember their actual names, so I'll just refer to them by these little shortcuts. And what they did is that they picked all trios and all pairs, all combinations, and they put them together, and they waited until the community stabilized. So basically, they did tons of experiments for very small communities. They even did for each pair and for each trio replicates in which always one species was very abundant in the beginning and the others were very scarce in the beginning. So this gives us plenty of dynamical data to work with as a constraint for inference of interactions. Right. Now, of course, these measurements are quantitative, right? And we need qualitative data. So we had to binarize these measurements. But as it turned out, all of the species reached roughly the same maximum optical density across the measurements. So we just decided to go with global threshold, just throwing global threshold. And then, again, these details of this are not super important. Basically, there is some data cleanup necessary because the steady state assumption we make. So since every experiment should end in a fixed point, this is a very strong assumption. So we had to clean out some inconsistencies in the measurements, such as if we observed a species to always go extinct, but then end just barely above the threshold, in case it started very abundant, we would discard this measurement as we are actually not sure if this is correct or if it would have continued to go extinct, etc. So what we are left with after the binarization is a set of such dynamical constraints where we say, well, beginning with some initial state, we eventually reach this other state, which is a fixed point. We can't go further. And this is for all possible pairs, for all possible trios. And we didn't make our own tool from scratch because Boolean networks are an established model. So we just used a tool that already exists for making analysis of most permissive Boolean networks. The trick is that this tool expects the interaction graph to be part of the input. So in gene regulation, typically the influences, the interactions between a transcription factor and a gene are already known, at least partially. So people would be able to construct these interaction graphs, and they would only be interested in the Boolean function. In our case, it's exactly the opposite. We are more interested in finding the interactions than the actual Boolean functions that explain the dynamics. So what we had to do is basically we had to do a complete graph and tell the tool that it can erase interactions that it doesn't need. And this is, of course, very, very expensive because there's 3 to the power of 64 possible interaction graphs that would match this one. And then, of course, on top of that, the number of Boolean functions, the monotonic Boolean functions with n on n components is a Dedekind number. It's the nth Dedekind number, and Dedekind numbers go quite fast. So I think for 8, it's roughly 10 to the power of 20, 25-ish. Right, so we are doing quite a brute force analysis here. But anyway, we were able to, for 8 species, still obtain some results. So the first and main result is obviously that we can enumerate the so-called subset minimal interaction graphs. So subset minimal here meaning that if we took any interaction out of this graph, it would no longer match the input dynamics. And by match the input dynamics, I mean there would be no Boolean network with this interaction graph that could replicate all our inputs. Now we found 4 subset minimal interaction graphs, which is surprisingly little, and all of them share 36 interactions. So we achieve very high precision for this particular application. You will see that immediately that I'm cheating here a little bit because I have these blue edges here. And I didn't mention this explicitly, but when the interaction is positive or negative, it means that the Boolean function is locally monotonic in its inputs. And we found that if we tried to do only locally monotonic functions, we wouldn't find any Boolean networks compatible with our data. So we had to allow some interactions to be non-monotonic. So in some cases to act as activation or to act as positive interaction, and in some other cases to act as a negative interaction. And that's in particular these two here. There are other interesting things that we found is, for example, that this node PCH is the most adapted species to the conditions in the lab because it doesn't have any incoming interactions. It's always one. It always survives. So it's kind of the community driver in a way. But yeah, so to explain why these two interactions have been chosen as non-monotonic, well, because our data comes from pairs and trios, we can do analysis not only for the whole community of 8 species, but also for any subcommunities. And indeed, if we look at subcommunities of size 3, we can find a conflict in the interactions. So you can see here that this one has a PV-SM interaction that is positive, but these 3 have PV-SM interaction that is negative. And these are the minimal subset minimal interaction graphs obtained for the community of these 3 species, right? So each of these trios of species, they only have one interaction graph that is subset minimal. And that kind of tells us that to observe the behavior we want to see on this trio of species, we would need a positive interaction in this case, but negative interaction in these 3 cases. So this is why we chose the PV-SM, right? So if I come back, this is the PV-SM. That's why we chose the PV-SM to be non-monotonic. And the second one was identified at subsets of the subcommunities of size 4. So here we have PA-EA positive, and here PA-EA is negative. It's the same principle. Now the small trick is that the tool we use, Bonesys, doesn't actually support non-monotonic interactions.
So
the reason,
sorry,
yeah,
Juri,
sorry.
quick question.
Yeah.
How do you define a monotonic interaction again? Or
Right.
how do you define non-monotonic interaction?
Yes. So non-monotonic interaction would mean that, let me go back here. So here, for example, there would be some configuration, some state in which if I increase PV, then SM would also increase. And there will be a different configuration in which if I decrease PV, then SM will increase.
I see. So you cannot define it in a strictly Boolean network, but once you have concentrations, then you can define it. Is that the idea?
No. So I meant, okay, so I'll rephrase. So there's a configuration in which PV is 0, and if I increase PV to 1, then the function of SM will be changed from 0 to 1 as well. So this would be the positive effect. And then there will be a different configuration. I can also say again that PV could be 0. And then if I increase PV to 1, then the function of SM will change from 1 to 0. So this will be the negative effect.
Okay.
And in this means that in the same function, you can find 2 instantiations, two configurations for which the function will behave differently depending on how PV changes.
I see. So the configuration is like configuration of the whole system. So this is some kind of nonlinear. Yes. So it's some kind of nonlinear thing then, right? Nonlinear interaction between PV and SM. And by nonlinear, I mean like SM and PV. It's not just them interacting. It also depends on the state of the rest of the system.
Yeah, of course.
Okay,
so here SM will depend on every single guy that has a arrow into SM.
Okay.
Right. So EA and PA and everyone else also has an effect on SM. So depending on how these other guys are arranged, PV can act either positive or negative.
Yeah, got it.
On SM.
Got it.
Yeah. Right. So as I was saying, Bonesys doesn't actually natively support non-monotonic interactions. There is a number of reasons for this. Mostly because monotonic, or locally monotonic functions are much easier to deal with, and in fact even have a lower complexity, analysis complexity when computing. So what we had to do is these little hacks here, that we would duplicate the nodes to create positive and negative interactions in parallel. And this interaction would basically be just copy the value. Now this small hack encoding is under some conditions it can create, it can cause an over approximation. So we would get some extra behaviors permitted simply because these two variables could become distinct, but it's under very rare conditions. So we hope that it's minimal. And in any case, we are already looking in a way at an over approximation of what can happen. So further over approximation doesn't devalue or doesn't invalidate our results. Okay, so another thing that we can look at beyond the interaction graphs themselves are so-called high-order interactions. So high-order interactions, in this case, this is a second-order interaction of some species C with the interaction from B to A. And these types of interactions have been identified to have very strong influence on shaping the steady state of microbial communities. So it's of course of interest to detect if any such interactions are present. Now, you may notice that in Boolean networks, we only have pairwise interactions. So this particular inhibition of inhibition can be abstracted by putting a direct pairwise interaction of C with A, but it comes with a caveat that in case B is not there, C should not have any effect on A, right? Because C only interacts with the interaction from B to A. So if B is not there in the first place, there should be no interaction from C to A. Now, this is something that, for example, the continuous models like generalized Otka-Volterra cannot express without structural modification, right? So I don't know if you are familiar with how generalized Otka-Volterra looks like. Basically, you have an influence matrix between the species, which is also pairwise, and the influences cannot be tied to one another. So you cannot say, oh, well, the amount of influence from C to A cannot exceed the amount of influence from B to A, etc. That will require structural modification. But in Boolean networks, because we have the different functions, we can express this as an OR in this case, which means that if P is 0, B is absent, then this literal is 1, and of course the entire function is 1, no matter what C is. So indeed, we are independent of C whenever B is 0. So searching for these kind of interactions, of course, requires that we do not look only at the interaction graphs, but we also look at the local functions. This is slightly too expensive to do on the level of all eight species. So we try to look at only the trios, so the subcommunities of size 3, and we were looking, can we find any second order interactions? So the conditions were that it has somehow the right shape here of the function, and that the second order interaction is not present if the first order is not present, right? So if we look at a community which had A, C, and D, then we wouldn't want to see the positive interaction from C to A. Now, we found few, and some of them were quite interesting because, for example, here we found 3 of them which share the target and which share the second order interaction. So this PCH guy can activate somehow these negative interactions from 3 different species PP. Now, these kinds of symmetries that we can detect in the interactions are quite interesting, I believe, because they can tell us that probably these negative interactions use the same metabolic mechanism, and that all these 3 species impact PP in the same way. Or that's at least the hypothesis we can generate. Okay, so our final result would be pertaining the stable states. So you may recall that the data we got, the paper that we got data from, the aim was to use, I hope I said this, to use the pair and trio measurements of stable states to predict the stable state of the entire community. And so what they did is that they also measured the stability of the entire community, and that's depicted here with the black boxes on the white boxes on the right hand side. So if you look here, this is the all 8 species. White means that the species went extinct, and black means that the species remained in that community. So they measured that if all species are put together, PCH, PF, and PV would be the stable community. And they did this also for all the subcommunities where one species was left out. So in this case, it was the EA was left out. In this case, the PA was left out, etc. Now, we did in a way the same thing. So we had our model that only considered data for pairs and trios, and we asked, okay, what are the attractors of the Boolean network? So in particular, the fixed points of the Boolean networks for this compatible with this data. And you can see that we weren't always hitting exactly the same. So in some cases we matched quite well. In some cases it's a little worse. But it's also interesting to note that if one looks beyond what's published in the paper and looks into the actual data from the measurements of Friedman and friends, you will see that, in fact, this, for example, in this delta PV, this stable community consisting of EA and PCH was almost always the result. But in some cases, in particular when the species PP was the one to start with high concentration, the stable community would be PP and PCH, and EA would go extinct. So actually, this gray bit here that comes from our measurements accurately predicts this alternative result in case a different initial condition is considered. So there is a possibility to predict the multi-stability here, multi-stationarity. But in some other cases, this is, sorry, yeah?
Just a quick question. So on the vertical axis, the deltas mean that if you don't have that species, right? Like you don't have EA.
Yes.
Now what's on the horizontal axis?
This one.
Yeah, what does that mean? So what does the column of PA mean, for example?
Yeah, so this means that if this species is present or not in that stable state. So, okay, I will explain from the start. So here, the black would mean that PCH is there in the stable community for the all taxa. And the white would mean that PA is not there. So PA went extinct, but PCH survived.
I see. And what are the colors then?
Yes, so the colors I use only to distinguish that, because we find multiple fixed points. So the colors are to just distinguish the different fixed points we find. So one fixed point, we find the EA, PCH, PF, and PV are all present. And the second one is the same, but we don't find PF. And the third one is only PCH and PV.
Okay.
So these are just different fixed points that we find. I apologize, I should have said this in the beginning. But yes, so these are just different alternatives of what we find in our model.
Thank you. That's right.
Yeah, right. And so as I was mentioning, in some cases, this doesn't have a very promising explanation in the data. In particular, we tend to let EA survive much more than it does in actual experiments. So clearly, there is some data cleanup still left to do that to refine the results we have so far. But in general, I believe it's quite promising. Okay, so this was to compute the fixed points or predict the stability of the community. Just also to explain these stars here means that the computation didn't finish in 48 hours. So we only have partial results. And this double star means that we actually found 10 different solutions. But this solution that is depicted is the only one that was present for all 4 of the minimal interaction graphs that we had. Okay. And that's actually all from me. So thank you very much for attention. I will, of course, happily take questions later. And I also want to thank some of my collaborators that have made this work possible. So Stefanie Wieder is an actual microbiologist, so she knows the little guys much better than I do. Loic is the person who spearheaded the most permissive semantics of Boolean networks and also the main author of the Bonesys tool. And Daniel is my boss. But he's always provides very valuable input and a very valuable motivation. So once again, thank you for attention.